My name is Manuel Copas, Dr. Manuel Copas, and I'm going to give you a research-based presentation on the latest bleeding-edge genomic analysis, the type of stuff that you will be exposed to if you decide to take the career of working as a biomedical scientist. So apart from being senior lecturer in genomics, I also run a new MSc degree in University of Westminster, which is on artificial intelligence and health data science. This MSc is currently the most successful MSc that we have, the most subscribed, and as you can imagine, in the type of area and times that we are living in, this could be something that is very much in demand. So all of the work that I do is computational. I don't do any lab work. And I've not done any lab work since I was an undergraduate, which is more than 20 years ago. And so all of the research that I do is on big data analysis. And we're going to focus today on big data analysis, specifically of genomics data, applied to human health. So hopefully by the end of this presentation, you will have a good grasp of some of the most challenging and exciting areas of research within the genomics area. So one of the things that I've been so passionate about, and the mission that I'm trying to work towards probably the rest of my career, is going about bridging the current inequities that exist in our understanding, modeling, research of biomedical science, which is mostly based on participants' histories, thinking of Northern European.
I don't think we have that slide.
Sorry?
I don't think we have like that slide in our the one our version.
No, you won't, because this is new. What you are looking at is probably last year's.
Oh, yeah.
Okay. So I told you that this is the very latest thing. So however, I will make sure that this is uploaded. Okay. These are fresh new slides, something completely different from last year. Hopefully that's something that you would appreciate somehow. And as I say, the main challenge that I am trying to solve is the inequities. Can someone think what's the problem if the data that you have from which you are basing knowledge is based on a certain population group? So what are the consequences of that?
Only work that group.
Whatever knowledge is derived from that group, is whatever knowledge derived from that data is going to work very well for that group. But the further you are in terms of genetics, in terms of environment, in terms of traits, the less accurate those current interpretations, those current models, guidelines, principles. You go to the GP, and then they're going to see, okay, so yes, I have this. If you are a male, 50 years old, and you have this hypertension, then I'm going to give you this amount of statins. But of course, you're not guaranteed that if you are female, 60 from South Asian descent, that same that same dosage is going to work. You are not guaranteed, and actually guarantee that most likely is you're going to need a different dosage. So that's the problem that we currently have. And of course, this is not a problem that that has been necessarily pursued. You know, why this is happening? Because obviously the research, the scientists, they were all white, wealthy British or Northern European people from the UK, from America, wherever, right? That's so that's kind of entrenched throughout generations of science and biomedical development, and that sort of that thinking will somehow have consequences. I mean, for instance, one of these anecdotes is that the aspirin, who has been prescribed for I don't know how many decades, they had never been tested in women until the 90s, something like that, right? So we just assumed that it would work, but we know that there are significant differences in our physiologies that can make things not work the same in males and females. I mean, that's very, very clear today, but it wasn't clear 30 years ago, 20 years ago. And we've been working towards augmenting the current lack of representation of specific population groups. But of course, producing the data, producing the clinical trials, producing all of the biomedical research that is required takes decades and is expensive, right? So how can we optimize the current resources, the current data that we have, so that at least we can identify where potential gaps in inequity gaps could be? I think that's one of the good ways to try to bridge the existing gaps that exist. So here I have this figure whereby 86% of all of genomic research participants that have ever been studied are of European ancestry. And yet, only 16% of the global population are of European descent. Okay, so now you can see this massive disparity in terms of the data representation. One of the analogies that I tend to put forward is that, you know, if you have a government, right, and you want in the government to have people, you know, politicians that represent your constituency. So if you have a constituency that doesn't have your politician, then you're not going to be represented where the laws are made, right? So that's the kind of similar analogy I can think of, right? So the consequences of these disparities are multiple. You can see, for instance, the fact that a lot of the things that we think are pathogenic, we then see, we have observed them in Europeans, and then I've seen the situation where we look at the same pathogenicity genetic markers in South American indigenous populations, and you see 20% of the population in South America for that particular pathogenic mutation, they carry it and they have no health effects. Okay, so a lot of the time we find situations where we kind of mistake what otherwise would have been noise as true signal, simply because we don't have representative enough data with which to make decisions. Does that make sense? Cool. And of course, the fact that we don't have all of the diversity that exist in terms of health, in terms of genetics, for the different populations in the world, that means that we are missing the richness and the potential mechanisms of how really physiology works. Because as I said, if you have partial or biased data sets, then the models are going to be partial and biased, sorry. Okay? So, and the problem is, now that we are in the high throughput technological biomedical science era with AI, if we leave things as they are, AI, rather than bridging the current inequities, AI multiplies them. Okay? So AI is everywhere. AI is not going to solve our problems. AI, what it does, it takes the data that we have and then it amplifies the power that we can do. So if you know this analogy I sometimes talked about, it's called the, you know, have I told you before this analogy of the sausage machine? No? Okay. So you like sausages. Some of you like sausages, right? So I had a friend who was a butcher, and he wanted to have the best sausages in London, right? And he spent £50,000 in the best sausage machine in the world. It had been tailored to his specific needs, shipped from China directly into his shop. And he spent £50,000 on it. However, he decided that actually, instead of using, you know, lean beef meat or pork or whatever, he wanted to use offal. Have you know what offal is? Like the sort of really the remnants of. So what happened is that he had the best sausage machine in the world, and he was putting rubbish meat. So what do you think came out? So you had rubbish sausages. It doesn't matter. It doesn't matter if you are spending £50,000 or you are spending £10 in the machine. If you put rubbish in, you get rubbish out. So AI is the machine, is the sausage machine. And the data is the meat that you put. So if you have rubbish data, what's going to happen? Rubbish models, right? Right? So what happens is that as a consequence of the existing inequities, these inequities with time compound. So have you ever invested, for instance, in ISAs or one of these accounts where they give you like these massive interest rates? You know about those, right? So what happens? You invest first year, let's say it gives you 10% interest rate, right? And you keep the money in. So at the end of year one, you have 110. Yeah? What if you had 100 before? Now, the second year, you reinvest the 110, and now you get 10% out of that. So what's going to be? No, it's not going to be 10 more. It's going to be 11 more because it's compounding. And so with time, you end up that you're not going to, it's not going to, the amount of interest that you earn is not going to be what you got at the beginning. It keeps compounding because it multiplies itself. And then, eventually it becomes exponential. So something like that happens when what we do in terms of models and our understanding doesn't change. So the whole point of this that I'm trying to make is that we need to have strong changes unless the current entrenched differences stop compounding. So unless we do radical change in a way, these entrenched inequities are going to compound with time, right? And I know that there's always a balance to make because, what you find is that, and you have seen it in research, right? We all end up listening or studying the same, BRCA1, BRCA2, the same genes, because those are the ones that have been studied before. So we then carry on and we study them more. So there is this sort of biomedical structural research practice whereby we keep knowing more about the stuff that we knew a lot before rather than new things, because since this is what we know, then we can get even deeper. And so what I'm trying to say is that there are big gaps that we don't even know about. And it's simply because to start with, the development of biomedical research has been skewed towards historical inequities. All right. So I think hopefully that makes a lot of sense, and hopefully this motivates the type of work that we should be developing in the future. So I'm going to be talking a little bit about the current state of the art on what we call precision medicine. And by precision medicine, we mean that we use big data to make decisions that are tailored specifically to the patient. So whereby rather than trying to generalize and having one size fits all, now we look at the individual, and based on the individual, we tailor the specific diagnosis, treatment, yeah? And each individual, depending on their genetics, is going to have their own expression of the disease, rather than having one specific disease that, in which we sort of pigeonhole everyone, right? So this is a kind of mentality. And we call it precision medicine because it's based on big data. But how can you have precision without being able to encompass all of the potential data that is out there with biased and partial data sets? So in a way, the sort of utopic vision of precision medicine cannot work if we are working with partial data, which is what we are dealing with at the moment. I'm going to be talking about diseases, and I'm also going to be talking about the implications of the current research. So before this thing of the Human Genome Project came along, we had this sort of what I'm going to call traditional medicine, whereby you basically prescribe or you diagnose based on the average, right? So we treat the average patient based on population-wide studies. And what happened is that you would end up having trial and error prescriptions and potentially adverse reactions. If you ever have, I would like you to stop talking. I would like you to stop talking, please. Or otherwise, please go. Yeah? Cool. So the trial and error prescriptions and adverse drug reactions would be based on, someone goes to the GP and they say, well, I'm just going to start with a little dosage and see if it works. And then little by little, they keep augmenting the dosage, okay, this doesn't work enough. Okay, so I'm going to try again. So this kind of trial and error is what I would call traditional medicine. And of course, It may work for the majority of people, but there's a huge amount of other people that for which they're different, or they don't necessarily are going to respond because of, for whatever reason, they're not going to respond the same. And of course, this means that we end up prescribing very inefficiently because we just don't have the adjustments for the people's genetics. And now we have on the other side of the slide, you see the precision future, where hopefully by looking at the data that you get from the patient, you can have this sort of all-encompassing vision of the right drug for the right patient at the right time at the right dose. And this is incredibly ambitious, and it takes a lot of effort and a lot of investment for this to happen. Okay? So for us to go from the traditional to the precision future, there's a massive gap that we can begin to address if we are more aware of the current limitations from which we start with working. There's this, not sure if you've come across this P4 medicine. Have you heard of this before? P4 medicine, so personalized, predictive, preventative, participatory, okay? So that's the utopic vision of having medicine that is tailored to the genetic makeup. We all know that we are 0.1% different genetically, and that difference makes a massive difference, right? We want to be able to treat patients before symptoms arise. By the time you have symptoms in a pancreatic cancer, you're probably too late, yeah? And you're probably gonna get into deep trouble by the time you have symptoms. So a lot of well, pretty much most of the healthcare that we get is reactionary rather than preventative. And so we want to be able to intervene early, because the earlier we intervene, the easier it becomes, and the less damage has been produced, okay? And we want to make sure that patients have an active role. In other words, they are not passive recipients. They participate in the decision making. They have a voice when they are being treated. So for every person currently, this is a Nature paper that came in 2015. I'm not showing the exact numbers, but basically they found that for every person helped by the top 10 grossing drugs, between 3 and 24 people did not improve. Yeah? So you can imagine the vast majority of the top prescribed drugs are having either no effect, or even worse, having adverse reactions in the patients. Okay? That's the vast majority of the drugs that are currently being prescribed. So this is just reinforcing what we said. So precision medicine focuses on individual responses rather than population averages, so that we can enable patient-centric diagnosis and treatment. So we move from one size fits all to tailored treatment based on your genetics, based on your own circumstances, the right drug for the right patient at the right time in the right dose. So you all should be familiar now with what genetics and genomics is. So genetics is the study of individual genes. So think about, for instance, studying single sentences in a book, versus genomics, which is the study of the entire genetic instruction, i.e. the whole genome, including interactions, environment, evolution. So genome is the complete organism's set of DNA. genetics is the study of individual genes, and genomics is the study of all genes. Can someone remind me how many genes we have? Well, it says there 20,000 protein coding genes, all right? 3.2 billion base pairs. We all have somewhere between around 5 million genetic differences between each other, right? And we are around 99.9% identical, regardless, okay? In fact, we are all Africans because we all come from Africa. So the Human Genome Project was released, its first version in 2003. And the way it works is that you end up having a Google map of the genetic data. So why is Google Maps so important, so useful? Because you have coordinates where you can map specific locations, functions, nodes, annotations into specific coordinates. So by having a human genome reference, you have the specific location of where each of the 20,000 protein coding genes are located. And that's very useful because by having these very well localized functional units, you can compare the differences between individuals at the level of the map, those coordinates versus what you see in their clinical history, in their traits, in their physiological expression, right? So that's really what clinical genomics is about. It's about understanding how the genomic differences that you see in the individuals affect their clinical lives, clinical traits, etc. All right. So the reference, the human genome reference, is the map against which we compare all other patients and find quote unquote variants. And by variants, we mean differences between the reference genome sequence, ATC, ATTTT, versus the sequence that you observe in the individual. So these are very basic type of variants that we have. So we have SNPs, or single nucleotide polymorphisms, which is a change in a nucleotide letter from A to T, for instance, and it's the most common type of variation. We also have indels, i.e. insertions and deletions, which are small chunks of DNA that have been added or removed from the sequence. And then we have copy number variations, which are large sections of the genome that are repeated or missing or duplicated, so on. So these are the three main types of genetic variants that you see in the genome. And here you can see how genomics has evolved in the past 20 years. So from the first Human Genome Project, which cost around 100 million in 2001, today you can have a whole genome for about a millionth of the price from what it cost 20 years ago, right? So the problem is that most of the data that was used to develop this genome reference was based on Europeans, okay? So these differences is what makes us look different, what makes us, some people have higher risk of cardiovascular disease, other people have higher risk of type 2 diabetes, other people have higher risk of developing congenital diseases in their offspring. So all of these differences, they have a substrate, a genetic substrate that for the first time we can identify like never before, right? And that is precisely what we are trying to work here. We're trying to understand and characterize how all of those small differences, the vast majority are neutral, but there's a small percentage that potentially leads to pathogenic mutation, pathogenic effects. And so we want to understand first, which of the variants that we observe are pathogenic, and second, how we can identify those so that if they are carried by someone, you can then do something about it. Even if you cannot cure, at least you can manage it, okay? So between 2008 and 2012, I worked at the Sanger Institute in Cambridge, which is the leading European genomic sequencing center. And I was developing a database called Decipher, and Decipher helps clinicians identify, by uploading their genetic data into the database, it helps prioritize mutations that are likely to cause disease in their patient. So what we realized is that those mutations, the vast majority, they are benign. Okay, the vast majority, they don't do anything. But there's a few ones that if you have them, they become terrible. Okay. So it was precisely around 2008 where massive parallel sequencing, next generation sequencing. Have you all heard of next generation sequencing? So it really started around 2008. And the point of next generation sequencing is that it allows us to read, really, to read the whole length of the DNA sequence in the patient. So we moved from the analogic, whereby you look at the karyotype on the microscope and you kind of see images, and then based on the shapes of the chromosomes, you say, okay, there's potentially a mutation here, there's not a mutation here, and so on. The problem with that is that the vast majority of mutations are much smaller than what you can detect under the microscope. And so by having next generation sequencing, now you have resolution at the base pair level, which means that you can identify every single variation. Just to give you an idea, one whole genome weighs around 200 gigs. Yeah, I'm going to say that again. One genome occupies 200 gigs. Yeah. Okay. How big is your Google Drive? I bet you it's one terabyte probably. No. How big is your Google Drive? I don't believe it's 15 gigs. It's more than that. 15 gigs is what you have in your iPhone. Now now we are talking, right? And I've given you a comparison. Yeah, you understand how much memory you have in your mobile phone, right? 15 gigs probably. And I'm saying that a whole genome takes 200 gigs. Okay. Now, you try to analyze by hand that. Yeah? Or try to download. How long do you think it would take to download 200 gigs? A whole night at least, wouldn't it? If you are lucky. If your brother is not playing Roblox or something. So can you see that we live in a different world? Yeah, you and I, we live in a world where, frankly, it's all about data, isn't it? So, and what you do with that data. So the key point, two key points. One is that it's rather accessible because, you know, $200, you can pay $200, right? You've paid that for, you know, a subscription to Tinder or something. Yeah, you know, no, it's not much. And then you get 200 gigs of your whole genetic data. Yeah, and that is basically telling you a lot about your own physiology, health, future health, right? You know, there are diseases, Alzheimer's, late onset Alzheimer's, that you are currently healthy, but then when you are 70, they start appearing symptoms, right? So, you know, at least one or two in this room will have late onset Alzheimer's, and you would be able to see that just by looking at your whole genome. But of course, I said it's 200 gigs. And then at the same time, that a lot of the data is basically based on biased references, and that a lot of what we know is actually wrong, and a lot of what we don't know, we don't know. Okay? There was this US politician who said, you know, we have like things that we know we don't know, but then we also have unknown unknowns. And that's the problem. Houston, we have a problem because the things that we don't know, this is the hard bit. And I believe that we can only get there if we have I don't have a solution, but certainly at least we should be aware that all the foundations that we have, although they are as good as they could be, they are a little bit shaky. All right. Okay, back to this thing. So the vast majority of the genetic data that we are able to identify in the 200 gigs, we have no idea what they do, what they mean, what they are. Yeah, the vast majority. You know why? Because in order for us to really identify every single possible effect, we would need almost an infinite amount of money. Because you need to develop a clinical trial, you need to have controls and patients, and then you have to have a lab validation. And as all of you are doing lab work, that's expensive. You know, you try to recruit patients. So it's a massive challenge. And when they tell you all of these things about medicine of the future and all of the things that are happening, hopefully you are getting a little grasp that not only the biology behind disease is incredibly complicated, and we have very superficial understanding, is that there's a lot more than we don't know, that most people really have no idea that we don't know. So there are two types, essentially, and this is a reductionist approach. There are two types of diseases. We have Mendelian diseases. By now, you may have heard of the Gregor Mendel laws. Yeah? Right. Can someone remind me what Gregor Mendel said? What's the main gist of Gregor Mendel?
The genes of your parents will be passed through the offspring.
All right. So Okay, so you remember the peas, yeah? We had peas, and we had green peas and yellow peas, right? And we knew that yellow peas were recessive, yeah, because we had two alleles, yeah, and you had to have the two recessive alleles for the yellow pea to be expressed. And so the idea that Mendel is the fact that we have the individual assortment. So the fact that the traits are independently inherited, and that they can be segregated, and then that you have the dominant and recessive. Yeah, so these are the main things about Gregor Mendel. And so the vast majority of this, I would say, I'm gonna say two outrageous statements now. First outrageous statement: every single disease you can think of have a genetic contribution. Yeah. If somebody throws a stone at you and hurts you, what's the genetics of that? Well, the person is psychotic, right? So they gotta. So that's, you see. So to the extreme, you could think that, right? Okay. So that's first outrageous statement, which is not outrageous, not as outrageous as I initially put it. Every disease you can think of has a genetic contribution. Now, does those genetics actually are they determinant? In other words, because you have that particular genetic load, does that mean you're going to develop the disease? Yes or no? No. Why not?
Like type of diabetes, you don't inherit from your parents, it's caused by your environment.
Exactly. So we have the environment. But coming back to the Mendelian diseases, right? So the vast majority of diseases. So we said that, I'm gonna make this claim, okay? All diseases are influenced by genetics, yeah? And we know that the laws of Mendel, they are the basis of genetics, right? So now I'm gonna tell you a contradiction. Most diseases do not behave, the inheritance that you have doesn't follow Mendel's laws, even though they're heavily influenced by genetics. What do you think is that?
Like not all diseases are autism.
Because most diseases are what we call polygenic, right? So, in other words, type 2 diabetes. How many genes do you think are involved in type 2 diabetes? Rough guess.
50.
Thousands. Give me another disease. Cancer. How many genes? Thousands. What do people die of? Coronary artery disease, inflammatory bowel disease, type 2 diabetes, breast cancer, and prostate cancer. These 5 diseases, 20% of all deaths in the industrialized nations contain any of these, right? 20%, right? 5 diseases. And they're all highly polygenic. In other words, the genetic contribution is distributed across a huge number of very small, tiny contributions from many different genes. That's why we call them complex diseases. And the thing is that Mendelian genetics only works when you are dealing with monogenic diseases. Can someone give me an example of a monogenic disease? Cystic fibrosis, sickle cell, Duchenne, I don't, yeah, sorry? Dwarfism is, if as far as I understand, is polygenic. Although I may be wrong, I'm not on compilation. Types, yeah. Does that make sense? So actually, genetics is complex, especially when you're trying to relate it to diseases. So one very used tool to understand the genetic contributions of complex diseases, we call them genome-wide association studies. Have you heard of that before? So genome-wide association studies is basically a statistical artifact that identify which genes are contributing to a specific disease. So in other words, it's able to give us the identification of specific genetic variants that are likely to contribute to polygenic traits. You know, not all traits are necessarily diseases. Breast cancer is a disease, but also, you know, height is not a disease, but it's highly polygenic. You know, another, you know, BMI is highly polygenic. So every trait you can think of that you can measure, most likely is polygenic, and the statistical artifact for us to understand the contribution of different genes towards a specific trait, we call it genome-wide association studies. And genome-wide association studies, what they do, they scan thousands of genomes where you have cases and controls to find variants, to find genetic differences between cases and controls that are statistically different. So if you have the same population, same group of people, same sex, same everything, and then you have people that have breast cancer versus people who have women that have breast cancer versus women that don't have breast cancer, and they all come from, you minimize all of the differences. So any difference that you observe between cases and controls at the genetic level, if they are sufficiently different, statistically different, then it's likely to be an association. It's likely to be associated to the presence of that disease. Does that make sense? So that's what we call genome-wide association studies. Here we have an example, okay? So we have cases that is individuals affected by the disease, and then controls, which are all effects identical, except that they don't have the disease. And so you genotype them, and then identify genetic differences that are significantly different between cases and controls. And that will help us identify whether a specific difference is associated to the disease or not. All right? But one thing is identifying genetic contributions. Another thing is identify which of those genetic contributions are actually causing disease or not. Okay? And this is particularly used in non-complex diseases. So in Mendelian disorders, like for instance, the ones that you've mentioned earlier, cystic fibrosis and so on. So the American College of Medical Genetics provides a scale whereby you can rank by degree of pathogenicity whether a variant is likely to be pathogenic or not, okay? And we're going to find that the variants of uncertain significance, VUS, rates are higher in non-Europeans due to underrepresentation in reference databases, which means that we have the limited clinical utility for these communities. We've mentioned next generation sequencing, and I said that you can have a whole genome for somewhere between $200 to $1,000. It occupies around 200 gigs of data. So there are two main types of sequencing that you can do. You can do exome sequencing. Have you heard of exome sequencing before? Have you heard of genes having two types of regions, introns and exons? So the exome is the sum of all of the exons from all coding regions, which is around 200,000. So if you just sequence the 200,000 exons in the whole genome, you're going to have an exome. And it happens that 85% of all diseases concentrate in exons, because these are the coding bits of the sequence that are going to give rise to proteins, and proteins execute the function. However, they only occupy 2% of the whole length of the genome. So only 2% of the whole genome is actually coding. And it happens that you can trade off between how much you spend. An exome is considerably half the price of a whole genome, and it doesn't occupy as much. So a lot of the time, particularly, for instance, in prenatal diagnosis, exomes are, recommended. Yeah. But of course, you miss important regions by not sequencing the whole thing. Okay. So with and without genomics, you're going to have trial and error prescribing. We're going to have one-size-fits-all approach with some exceptions, some guidelines that your doctor might have, but really based on years of experience rather than any kind of methodological, systematic characterization of biological differences that come from the millennia of accumulation of mutations. People who lived in the Tibet versus people who lived in the Sahara Desert. They've been living there for millennia. They've adapted themselves to the environment. The Western people, we have a lot more production of amylase. Yeah, the enzyme that degrades starch and we have it in our saliva. Westerners tend to have tend to be able to drink milk. Many people outside of the Western world, they don't have the capacity of drinking milk because they don't have lactose, then they have lactose intolerance, right? So all of these things, unless you systematically characterize and identify them, you're going to end up having a very sketchy medicine, right? Whereas if we use genomics, we can have much more evidence-based selection, targeted, stratified. We're going to have high-risk individuals. We're going to have, specific, for instance, there's I keep mentioning South America because I have worked in South America quite a lot. So there's a indigenous community called Uros. They live on Titicaca Lake. Okay, Titicaca Lake is 4,000 meters high. Yeah, and I'm not sure if you've ever been skiing. Have you ever been skiing? Right. What happens when you're skiing? You have to put a lot of sun cream because the rays of sun, reflects on the snow. Anyways, and you are very high, so there's a lot of more ultraviolet radiation. So I went there. I was there 2 days. I put 50 factor sun cream, and I got burnt. And people there, they have no sun cream, nothing. They're dark, and they don't have more melanoma than I know. So there's something there, these thousands of years of adaptation that leaves a residue in our genomes that we're just beginning to understand that they exist, let alone use them for the purposes of benefiting not just them, but the whole of humanity. Because when we understand that, it doesn't just help them, it helps everybody else. The ones that are represented and the ones that are underrepresented. Because it helps us understand the real mechanisms underlying the biology that we observe, okay? So the global genomics landscape is that most of the biobanks, have you heard of biobanks? The UK Biobank, there are lots of big national initiatives that they take the NHS, for instance, and then they want to combine it with the genome data from the British population and then put it in a massive database. So these biobanks are the workhorse of the medicine, of the biomedical research that we are developing, okay? And there has been a big number of different countries, but the reality is that there's this 1.5 billion people in the global south who's never been in a biobank, who's being affected by diseases that are not even present by affected individuals in any of the existing biobanks. And hence, we still call it precision medicine. Yeah, so I insist you're never going to have precision medicine with partial data. So the UK, for instance, is now invested £105 million to perform the most ambitious newborn screening sequencing in the world, 100,000 newborns. That's going to be incredibly rich, not just for the UK, for everybody else, but simply because the UK is not representative. I mean, London is in a way, but the UK in general is not representative of all of the diversity and the richness of the world, even though London is exceptional, but it's not going to replace people who are living in different environments. So this is something that we really need to take into account. At the same time, bridging the inequities that exist in the representation of the data is not just about augmenting the diversity of the different populations. I think the UK, for instance, is incredibly good at making those data sets, biased as they are, accessible to any legitimate researcher in the world, as long as they sign a contract and they follow the principles of protecting the identities and the individuals who donated those data sets. So there are many different types in terms of the democratization of access of data, that even though those data sets might be biased, still making them accessible contributes towards a more equitable precision medicine. All right? This is just more of what I just said. Well, I've said that 86% of genome-wide association data comes from European ancestry, and that's problematic because it only represents 16% of the population. I've also said that the reference genome and our understanding of the effects that we see between genome data and biomedical annotations as a consequence of that data is primarily based on European data sets. The variants that we don't understand what they do are more common in non-Europeans. We find that most of the models that we have for representing complex diseases is based on European data, meaning the farther away you are genetically from European populations, the less precise those models are going to represent the genetic risks that you can currently identify based on those models. And therefore, we are missing discoveries that could potentially help us not just protect those who are affected, but also for us scientists and for the world to really understand the actual mechanisms of how genetics and biology work, because we will have the real sample of data. So if we think of genomic medicine as a subset or really the peak of the iceberg for the precision medicine mountain, so the challenges that we currently face, all of us, obviously we have an aging population with increasing chronic disease. I think at the rate at which we are all living now, we are all going to have chronic diseases. Yeah, unless we have an accident or something, we're all going to have chronic diseases. And yet the amount of money that is being invested in the NHS, as you all know, is flat. Then workforce gaps in expertise. Most of the people that work in these fields are not representative of the global population. Then, as I said, persistent underrepresentation, the fact that data governance, i.e. how we share, who has access to the data, who is the custodian of those data sets, are incredible challenges that we need to take into account. The fact that who has access to this data, because of course it's incredibly sensitive data, and you know, if your health insurance gets this data, maybe they might discriminate against you. If your employer gets this data, maybe they might discriminate against you because you have imminent cancer of stomach cancer or whatever. So this is incredibly challenging. And I've already mentioned that. So I'm going to be talking about some of the research that I've been conducting in Peru. We call it the Peruvian Genome Project. Peru actually has one of the most diverse genetic ancestries in the world outside of Africa, because Peru itself has most of the country on the east side is Amazon. Then you have the center chain of mountains and Andean mountains that go up to 6,000 meters high. And then you have on the western side, it's all desert. So you have Amazon, high mountain, and desert. And so it's incredibly varied. And in fact, I was surprised to learn that Peru has the greatest number of indigenous population in the whole of South America. About 6 million people, indigenous people, live in Peru, more than Brazil. I thought of Brazil being so big, but actually Brazil has something like 3 million people, indigenous people, even though it's like 4 times the size of Peru. But Peru has 6 million indigenous population. And because of the variety of different climates, geographical features, makes it incredibly diverse. And it's one of the most diverse places on Earth in terms of human adaptations. Part of my research work is to develop what I call the HEIM framework, Health Equity Informatics Metrics Markers. The idea is to quantify and score data sets based on the diversity and representativeness. I said that all of the reference data sets that we use, you know, the databases that clinicians use for assigning a diagnosis, they're all based on skewed genetic data sets. So we are developing a framework that helps score data sets based on their diversity and representativeness. Okay? And pharmacogenomics, which is the science that predicts your optimal drug dosage based on your genetics. Whenever you take a drug, the actual active principle that has the effect is not the one that you swallow. You swallow a pill that then gets metabolized, and that metabolized drug it has its effect because you have enzymes that are the ones that degrade or metabolize that specific chemical. So because it's carried out by enzymes, enzymes are proteins, and proteins are encoded in your DNA. So depending on the variation that you have on your DNA, you're going to have differences in how good, bad, or fast or slow you are at metabolizing a particular drug. And it's incredibly important because pretty much all of the prescribed drugs are affected by your genetics. And this is a very quick way to show actionable outcomes from sequencing your DNA. You might have heard of liquid biopsy, which is the detection of tumors, circulating tumor DNA into the bloodstream. They are non-invasive, real-time monitoring of treatment response and detects relapse months before imaging. You may also have come across something called direct-to-consumer genetic testing. Do you know anyone who has performed a genetic ancestry test? Have you done that before?
Don't trust.
Sorry?
I don't trust.
Ancestry is quite precise, actually. The one I don't trust so much is the ones that you buy that they predict your health traits. So that's not so good. But our ability to detect ancestry is incredibly accurate because there's a tremendous correlation. There's a massive correlation between your genetic variations and the geography of your ancestors. Okay, there's a massive correlation between the geography of your ancestors and your genetic code. Okay, and we can actually analyze and report on that. The most important currency that we have in healthcare is, like everything else in this AI age, is trust. Yeah. The currency of trust. If your credit card is stolen, you can get a new one. If your genome is leaked, you cannot change it. If you give your genome to someone, you cannot ungive. They cannot give it back to you. Okay. You cannot unknow what you already know. All right. So who owns your data? Can insurers use it against you? How do we share data for research? Anyway, so some of you wanted me to finish early, so just a couple of slides to finish. Hopefully, I've given you a slightly different view of genomics. Hopefully, maybe not, but that's my vision. I like to think that some of you have given you food for thought. Here are my key takeaways. So genomics is transforming medicine. But one thing, being a genomicist myself, they are not a crystal ball that they're going to tell you the future. They are not deterministic. Genomics really, if anything, knowing it can help make your future evitable. Yeah, so you can avoid future outcomes, perhaps by knowing your genetics. The interpretation of genetics is incredibly complex, it's incredibly skewed, and we need to have awareness of the limitations, especially for the burden of uncharacterized variations, especially in diverse populations. We have seen that most diseases people die of are complex diseases, and they require different frameworks to the traditional Mendelian genetics, simply because most diseases are the consequence of contributions from many genes. And finally, I like to say that equity is not optional, not even for the well-represented populations, because by having more representative data sets, everyone wins. We have a much better and accurate understanding of the real biology. And secondly, we make sure that precision medicine is what it's meant to be, which is inclusive for all. So thank you for listening. And I'm going to leave it there for today. And yeah, I hope that was useful.